So when we talk about the GENIE test, we're not talking about genetics, okay? When we talk about genetics, we're talking about the study of gene presence. What does that mean? Do you have a gene or not, right? And so it's sort of like, right, and when we talk about genes, we're talking about DNA. I'm showing you here. And that's essentially the instruction manual of your body, right? It's the DNA is what contains all the instructions that are going to be deployed so that you can have a functioning body. There's a process in order to get those instructions deployed, and we'll go over them. But when we talk about GENIE, right, we're not talking about genetics. And examples of something that has to do with genetics would be, right, doing our blood work for CIRS susceptibility when we're looking at our HLA haplotypes. What we're looking at there is, do you have a gene or a set of genes that are associated with susceptibility for CIRS? Another example is the ApoE genetic mutation, which has been tagged as, right, those people will have increased susceptibility to Alzheimer's. It doesn't mean they're going to have it in the same way that it doesn't mean you're going to develop CIRS. It just means that you have a higher risk of developing these issues at some point in your life. Same thing goes for the MTHFR mutation, right? We think about there are certain mutations there that have been identified as problematic when it comes to things like methylation and ultimately things like detoxification and things like that. So that all has to do with genetics, right? That's not what GENIE is about. GENIE is about what we call transcriptomics. It's a very fancy term. All that means is we're studying gene expression or gene activity, right? And I'm putting here in green what we're looking at when we look at GENIE. So essentially, how are your genes expressed? Expression takes place during the transcription of genes, in other words, from DNA into RNA or mRNA. M stands for messenger. You can think about transcription as converting a book, like a physical book, and that would be the DNA, into an audio format or a digital format. That would be the RNA, right? So that's what's happening here. So the instruction manual needs to be converted into something else, into another format that can then be utilized for a final output. This is what GENIE is measuring. It's this transcription process which reflects how our genes are being expressed, right? And gene expression can fluctuate, and I'll touch on that in just a moment. And as I said, RNA, which is this guy down here, is the messenger that is translating genes into proteins, which is the final bundle, the final output. That's what we're doing here. We're going from DNA to RNA to protein. And in this context, the analogy that I like to use is, right, in transcription, we're talking about converting formats, right, or modalities. Here we're translating, it's literally like we're translating the book from English into Spanish. So we're translating this into a new language. Like protein is it's like Spanish. It's like a different language from English. It has its own rules. It has its own world. So that's what RNA is doing. So you need that gene expression, and that gene expression is going to be the thing that translate things into protein. And what are examples of proteins? Well, we actually know these things. These are the things that we usually measure in our blood in LabCorp and Quest testing. So things like MMP9, VEGF, TGF-beta1, C4a. When we measure antibodies for tick-borne infections or for autoimmune markers, enzymes, and most hormones, all of those are proteins, and they're the byproducts of this process of transcription and translation. So I wanted to give you that little bit of piece so that you understand, right? When we're measuring a lot of this blood work and lab conquest, we're measuring at the level of proteins. When we're looking at HLA susceptibility, we're measuring at the level of DNA. When we're measuring GENIE, we're measuring at the level of RNA, a transcription, gene expression, which is here.